Hello guys, welcome to Life Science with Felix. Today we're going to be doing DNA code of life. Now doing DNA code of life, but before we start with actually DNA code of life, I'd like for us to do the structure of a cell. It is important to know the location and the function of certain organelles that are illustrated in front of you. A cell, it is made up of ribosomes, cytoplasm, chromatin network, nuclear pore, and the nucleus. This is how a basic structure of a cell looks like, and we're going to be moving into the functions of these organelles that are in front of you. Now, the cytoplasm is the body of the cell where metabolic reaction takes place and organelles of the cell are suspended, right? And then the ribosomes are the small organelles in the cytoplasm, which are the site of the protein synthesis, and the nucleus controls all the activities in the cell. And this is how a nucleus looks like, right? And the nucleus is made up of nuclear membrane, nucleolus, chromatin network, and a nucleus nuclear plasm, and the nuclear pore, right? We'll be looking at the function of those label part of the nucleus, right? The nuclear membrane encloses the nucleus, and the nucleolus contains three nucleotide bases and it produces ribosomes. The chromatin network contains DNA which forms chromosomes, and the nuclear pore it is a small pore that allows substance in and out of the nucleus, right? So now, since we've got that understanding of the cell, we've revised a little bit of the cell structure. Now we'll be looking into today's actual topic, which is DNA, right? So now we're going to be looking at the structure of nucleic acid. There are two types of nucleic acids in a human body. We've got the DNA and the RNA. So now the DNA in its full term, it means deoxyribonucleic acid, and the RNA in its full term, it means ribonucleic acid, right? DNA and RNA, both of them consist of the nucleotides, but in today's topic, we are going to be mainly focusing on the DNA rather on the RNA, but I will be doing more videos explaining the RNA and other parts of nucleic acid, what happens in the topic of DNA code of life, right? All nucleotides are composed of three compounds, which is the phosphate group, the sugar molecule, and the nitrogenous base. This is a simple structure of how a nucleotide looks like. It has the NB, which is the nitrogenous base, the S, which stands for the sugar molecule, and the P, which stands for the phosphate group. Now, DNA, it is found in two locations in a cell: in the nucleus, which is referred as the nuclear DNA, and outside the nucleus, which is referred as the extranuclear DNA. Now, with the extranuclear DNA, we've got two types of extranuclear DNA, which is the chloroplastic DNA, which is only found in the chloroplast of a plant cell, and we've got the mitochondrial DNA, which is the DNA that is only found in the mitochondria. Now, in front of you, you've got a very small illustration of the location of DNA. As I said, it can be found in the nucleus and outside the nucleus. The location that you see that are outside the nucleus, it is the chloroplast and the mitochondria. Remember those ones, they are called the extra nuclear DNA, which is the chloroplastic DNA and the mitochondrial DNA. In front of you, what you're looking at, it is the structure of a DNA molecule. Please take time to understand this structure and just to learn it, familiarize yourself with it. But I have taken some notes, written it down in the most simplest form for you to understand this structure of a DNA. The DNA has a double helix structure consisting of monomers that are called the nucleotide, which links to form a long chain. That long chain is called the polymers, and the sugar that's in the DNA is the deoxyribose sugar. The sugar is attached to the nitrogenous base, and the phosphate and the sugar molecules are attached to form a long line. So this structure, it's basically what I've just said. You can take time and just pause this video and take down some notes, guys. Continuing with our today's topic, as we said that the DNA consists of a nitrogenous base. These are the four types of the nitrogenous base that are in the DNA. The arrow that you see that's pointing at the lines is what we call the weak hydrogen bond that exists in the nitrogenous base. Right, in the nitrogenous base of a DNA, we've got adenine, thymine, and we've got guanine and cytosine. These bases, they are always complementary. They always pair with each other in this sequence that you're seeing. Adenine will always pair with thymine. Guanine will always pair with cytosine. Now, as I said before, the nitrogenous bases are complementary and will always form in a specific order. As I said before, adenine will always pair with thymine, guanine will always pair with cytosine, and they are held together by a weak hydrogen bond. The weak hydrogen bond that I was talking about earlier on, it is the one that you see in front of you, the arrow that points. This arrow that points at the lines, that's the weak hydrogen bond that exists between the nitrogenous bases, which it helps to hold them together. Right, now we're going to move to the function of the DNA. The DNA, its function, it is to control the functioning of the cell, to regulate the functioning of the genes, and also to pass on hereditary characteristics. So now I'm going to move to another topic that's in DNA, which is DNA replication. Right. Let me just open up just a little bit here. What's going on with this? Okay. All right, we're good now. As I said, we're going to be looking at DNA replication. DNA replication, it is the process in which the DNA makes an exact copy of itself, an identical copy of itself. At first, there is one DNA molecule, and after the replication, there is two molecules which are identical. That's DNA replication. That is DNA replication. Take time, as I said, take time. Pause the videos. If you don't understand, rewind, start over, and listen again. Now I'm going to be explaining the process of DNA replication in simple steps. Right? Now, okay, what's this thing doing now? Okay. As I said, I'm going to be explaining sorry about that. I was just having some technical problems. As I said, I'll be explaining the process of DNA replication. Now, at first, the strands will unwind, then followed by the weak hydrogen bond which exists between the nitrogenous base will break and the strands will unzip. Then each strand acts as a template for the formation of a new strand. As you can see, we've got two templates. We've got the first one and the second one at the bottom. The first one is above, and then the second one is at the bottom, and in between those templates we have what you call a free DNA nucleotides, right? The free nucleotides build DNA strand on each original DNA strands, attaching their complementary nucleotides, meaning that adenine to thymine, guanine to cytosine. So the summary of this process is that the DNA, which is in a double helix shape, unwinds as well as unzip to form two separate strands. Both DNA strands serve as a template to build complementary DNA, which is A to T, C to G. That is thymine. Oh, sorry, that is adenine to thymine, cytosine to guanine, using free nucleotides from the nucleoplasm. This results in two identical molecules, each will be consisting of one original and one new strand. Right, now we're going to be moving at the significance, the importance of the DNA replication. Significance simply means importance. It's the synonym importance. It is the synonym of significance, right? So don't get yourself confused when you see this word. It means significance. It means it means importance. Now the importance of DNA replication is that DNA replication, it is important for cell division, which exists in mitosis, and also it ensures that new daughter cells produced have the same number and equal chromosomes as the parent cell, right? That is the importance of DNA replication. Now, DNA replication, it only occurs in the interface of mitosis, or rather you can say the interface of the cell cycle in the nucleus. That's it for today's topic, but stay tuned for more videos that will be explaining this topic even further. As I said, I will be explaining the RNA. I'll also be explaining DNA profiling and protein synthesis. That's it for today.